Now that the background information is out of the way, we can get on to the actual heart of this course, which is working with ecological community data. Today I'm going to be talking about some common patterns that you see in ecological community data. There are two properties of species that drive biodiversity patterns. One is the occurrence of species, in other words, their distribution, and then the other is their abundance. And those are two interrelated factors that ultimately define biodiversity. These properties often respond in tandem to generate patterns that have been documented in a wide variety of regions and in a lot of taxa around the world. Several of these patterns have a biological basis. First off, it's been noted that most species are rare, few are common. And this is true both in terms of occurrence. In other words, most species only occur at a few locations. Only a few species are very widespread in distribution. And it's also true in terms of abundance. That is, for most communities, most species are represented by only a few individuals. There are very few species that have dominated the community in terms of number of individuals. The second pattern is that most communities will contain only a subset of what species occur within that region. So not all species present within a given region, in other words, the region's species pool, will be present in any given community for a variety of reasons that may relate to resource partitioning, for example, resource limitation, dispersal limitation, biotic interactions, or chance, or all of the above. And so therefore, what you'll see is a representation of what could be there. Furthermore, common species tend to be widespread, whereas rare species tend to be restricted in distribution. They have smaller distributions. So in other words, there tends to be a positive relationship between occurrence and abundance. And finally, communities that have high species richness also tend to have high overall abundance. In other words, those communities that have high species richness also have lots of individuals present. And again, this could be due to a variety of biological reasons. But there are also patterns that exist in community data that are a function not of nature, but of sampling. And one of the best known ones is that species richness is positively associated with the number of samples collected. In other words, sampling effort. So today's lesson is going to explore these patterns. If your data show patterns that are contrary to these standards, then you need to determine why. It could be something as trivial as an error in data transcription. It could be due to low sample size, or it could be something real and odd about your system. And all of those are things you need to know. So once again, we'll be working with the Bryce Canyon vegetation data set. So you will read that in. And then what you're going to do is you're going to use the package labdsv. And in it, it has a function called ABUOCC, standing for abundance and occupancy. And by applying it, it creates 4 graphs that illustrate patterns of abundance and occurrence. And we'll go through all 4 of those in sequence. The first graph that is created is this one. It shows on the x-axis species ranked from most frequently encountered to those found at only one site. So that's what it means by species rank. And what you can see here is that most species occurred at only 20 or fewer plots. So most of the sites here indicate that there are relatively few species at it. And so hence, this graph illustrates the pattern that most species are rare, few are common. And this is true of occurrence. So most species occur at only a few locations. Only a few are more widespread in distribution. So this is also a truism for abundance, where only a few species will comprise the majority of individuals. So I'll show the abundance patterns on the next slide. This graph plots species on the x-axis, ranked from those with the most individuals, near the origin, to those with the fewest, down to species represented by singletons, meaning only one individual. And then the abundance of each species is plotted on the y-axis. So the difference in this graph and the previous one is in the dependent variable, which is the y-axis. The previous one was occurrence, whereas this one is abundance. This type of plot is called a rank abundance curve, or a dominance diversity plot, or a Whittaker plot, and I've got some other synonyms in the lecture notes for today. The shape of the curve, its slope, can indicate certain features about a community, and different communities can be compared by comparing the shapes of their rank abundance distributions. This one I've fitted with a log normal function, that's the line. But there are over 24 shapes that species abundance distributions can take, each resulting from a different process. We will cover much more about this topic in an upcoming week, so I don't want to go into much detail about it here. I just wanted to illustrate the point that in most communities, species have unequal and usually quite skewed distributions. I just wanted to cover this here for the sake of completeness about this discussion of rarity. There is rarity of occurrence and rarity of abundance. And speaking of rarity, Deborah Rabinowitz came up with what she called 7 forms of rarity based on 3 traits. One of these is the geographic range of a species, in other words, its occurrence. Then also on the population size of a species, in other words, its abundance, and then on its habitat specificity. And based on these 3 traits, she came up with this table that illustrates different species in terms of their degree of rarity. And those species denoted with these pink boxes here would be considered rare in some way. And so again, to reiterate, rarity is common, commonness is rare in ecological communities. This graph plots the number of species on the y-axis versus plots, or sites, ranked from the most speciose to the least on the x-axis. And by speciose, I mean species rich. And you can see that most plots had 15 or fewer species out of 169 total in the Bryce-Veg dataset. So not all species present in a given region, or part of the regional species pool, will be present in any given community for a variety of reasons, such as resource limitation or dispersal limitation or other factors. So most communities will only contain a subset of those species that could occur within that region. Nestedness is a special manifestation of this relationship in which less speciose sites consistently contain a non-random subset of species from the overall pool. It has been used as an indicator of biotic mechanisms governing diversity, such as mutualistic relationships or competition. Now, this figure isn't quite right for our situation, since all of the sites in Bryce Canyon, the Bryce Canyon vegetation dataset, were collected from plots that were the same size. You may recall that each was 375 square meters. So this figure with these patches of different sizes isn't quite right, but it shows nestedness, it illustrates nestedness in a nice way. But even though our plots were the same size, they contain different numbers of species. So we can assess whether or not there is nestedness present in the Bryce Canyon vegetation to see if there's a non-random association of species. And this is one way of doing this. I'm not going to get into this. This is itself a huge area of study. And so if you're interested in this, we can talk about it more sort of one-on-one. So anyway, this is one way of doing it. And this is the kind of graph that you get. To interpret this graph, what you see here is there's going to be a line, and then there's going to be things that are representing omissions and commissions. In other words, unexpected absences or omissions, and then unexpected presences or commissions here. And so if we go back to our data here, you can see that there really isn't any nestedness going on. There's lots of omissions and commissions. So these data do not appear to be nested, at least by this measurement. This nestedness stuff is an example of a kind of rabbit hole that you can really go down in exploring patterns of occurrence in your data. So to show the original plot, once again, our data aren't nested, but each site is only a sample of the overall species richness. And in our case, it is a random rather than a nested sample. Common species tend to be widespread, whereas rare species tend to have smaller distributions. In other words, there tends to be a positive relationship between occurrence and abundance, with widespread species occurring in higher densities, in other words, more individuals, compared to species restricted in their geographic distribution. If you were to fit a regression line through these points, you would see that it has a positive slope, indicating that the most widespread species, those that occurred in the greatest number of plots, were also the most abundant ones on average. Even without such a line being present in this graph, though, you can still see that the rarest species, those with the lowest abundances, were found in the fewest number of plots. This graph illustrates that those plots that had the most species also had the most individuals present in them. So all of the previous graphs illustrate patterns very commonly seen in community data that are due to inherent factors about the species themselves, and they all have a biological basis. Now let's explore a pattern that emerges as a result of sampling. Another pattern that is seen in ecological community data is that species richness is a function of sampling effort. In other words, species richness is positively associated with the number of samples collected because of the increased likelihood of encountering a rare species with more sampling. So, a species accumulation curve allows you to determine how much sampling effort is needed in order to adequately represent richness in a community and whether your sampling has done so. And you do this through presence of trying to detect the presence of an asymptote. So these kinds of curves or graphs are sometimes called collector's curves or sampling effort curves or species discovery curves. I tend to use the phrase species accumulation curve. And the simplest type is shown here. It just plots the raw numbers of species as a function of the number of samples collected. And this results in what's called a collector's curve. This graph in particular, you can see an asymptote here. And it indicates that only about 16 species were needed in order to fully represent all of the species present in this community. If you don't find an asymptote, in other words, if the curve is still increasing, then that means that you have not represented all of the species that are potentially present in your community, and you need to continue sampling. Here's a different way of presenting a species accumulation curve on the same data, but this time this one uses error bars and it smooths out the curve. And this is called the exact method. And what you do is, again, you look for an asymptote in the error bars here, and you can see that they level off, once again, about 16 species, consistent with the previous curve, which is expected since they were based on the same data. Now, if you have different habitat types, and you suspect that perhaps species were easier to sample in certain habitat types than in others, maybe species are easier to hide in certain habitat types than in others, then you need to do this kind of plotting of a species accumulation curve for each one of those habitat types. Like here. From this graph, you can see that fescue had more species than mixed grass communities did. And you can tell that because the fescue line is higher up on the y-axis than the mixed grass one is. Even though there were fewer sites sampled, fewer samples from fescue-dominated areas, and you can tell that because it has a shorter range on the x-axis than the mixed grass samples had. So there were more samples made in mixed grass dominated sites than in fescue dominated ones. The curve for fescue doesn't show a strong asymptote, suggesting that there are likely even more species present in fescue dominated areas that you haven't represented in your samples yet, and so perhaps more surveying is warranted in fescue dominated areas. The curve for mixed grass appears to be leveling off, indicating that the species richness value from it is likely correct and that it wouldn't change much with more sampling. So for your assignment, you're once again going to be working with the ground beetle dataset. For the past couple of weeks, you've been working with the ground beetle's habitat.csv file. It's a site by environment dataset. And in a few weeks, you're going to be working with the ground beetle abundance file. It's a site by species dataset. And these two files express data in a certain way. This week, you're going to be working on the same data, but with files that express those data in a different way. And so you're not going to be working with these, you're going to be working with these files. So GB BIOL and GB site. GB BIOL is a presence-absence site by species matrix, whereas its sister file, the ground beetle's abundance one, is abundance site by species. And then GB site is a pared down version of the ground beetle's habitat site by environment data set. It's got just 3 columns of information on the 18 sites, habitat type, and the maximum height of vegetation at each, and nothing else, whereas the ground beetle's habitat file has more. It has rows of sites, which it calls samples, rather than rows of species like in the GB BIOL or the ground beetle's abundance files. It is typically much easier to use Excel to create alternative forms of a data set than to try to wrestle with it in R. And since this course isn't really about R, we will use these alternative CSV files rather than spending time trying to manipulate things in R. Okay, so last week you addressed the most basic questions about any ecological community. What is the overall species richness, and which habitat type had the most species? But without an assessment of species accumulation, the values of richness that you obtain may be underestimates if your sampling hasn't been adequate. And so, using the skills that you gained today as well as over the past few weeks, you're going to address the following questions that pertain to abundance and distribution. Namely, you're going to find out if the ground beetle data exhibit the common patterns that are typically seen in most ecological communities. Are rare species common and common ones rare? Is per site richness only a subset of overall richness? Is there a positive relationship between occurrence and abundance? Do sites with high richness also have high abundance? And then finally, was sampling adequate for each of the 3 habitat types? In other words, there were 6 replicates per habitat type. Were 6 enough? So today's lesson focuses on describing fundamental properties of communities, teaching you to recognize some patterns that are repeatedly found in community data. Now that you're aware of these things, you can detect whether your focal community is behaving normally or not, and investigate accordingly. The next 2 lessons will focus on how to quantify foundational community structure in terms of species diversity as a function of richness and evenness and patterns of abundance.